onward and upward. Here we're going into, we just finished steroids and the lipid category. So I just to link those videos together, here's what you're looking at. So we're seeing two standard kinds of lipid or steroid structures that you see. But now we're really into probably, I would say, the meat of the matter, but that's a bad pun. This is proteins. And proteins are the most abundant, the most, in some sense, significant molecules that we're going to talk about for a good part of the year. And that's because proteins are us. We are made of protein. They have all of these and more of these types of functions. So enzymes, we've mentioned already, they are proteins. There are proteins that transport materials. There are proteins that protect us in our immune system. There are proteins that form basic structures of our body, including muscles. There are proteins which regulate the things that come in and out of our cells. And there's some pictures over here on the right, and this is not OpenStax. I just took this from somewhere else, but it just gives you an example of the wide range of things that proteins can do. So you can take a look at those pictures at your leisure. Here's some very specific ways and examples of proteins. So here's one example, digestive enzymes. We said enzymes are proteins. We mentioned that in the part on carbohydrates. When you can see that their importance is to help move chemical reactions in a positive direction more quickly. So they help to speed these things up. They serve as catalysts. They catalyze reactions. Transport. These proteins here, hemoglobin is part of our red blood cell. It's a protein involved with carrying oxygen. Albumin is another one that is involved with carrying other dissolved materials, and we'll talk about those maybe. Structural, some proteins serve to act as basic structural components for lots of different things that we know of, including these areas that we're going to learn about called the cytoskeleton. There's actually sort of a skeletal framework that makes up and supports cells. Hormones, insulin and thyroxine, do all sorts of stuff. Defense, proteins such as the immunoglobulins are part of our immune system. Got carried away. They're contractile proteins, which help muscles to move, and storage proteins, such as those which will hold on to energy and help something develop. So in the little seed observation we made, basically what we see in the interior of a seed or an egg is some storage material, including proteins, that will allow, when that seedling or little organism starts to grow in the early stages, has some ready-made energy supply for them, as well as some raw materials for making structures. So we'll take a look at some of these in a little more depth. Enzymes we'll talk about a lot, but enzymes are super important. They're proteins, and enzymes can do lots of stuff. They can make reactions happen faster that break down materials. We talked about that in digestion. They can also aid in the building of new, more complex structures. So something that breaks down is called a catabolic process. Something that builds something up is called an anabolic process. You've probably heard of people using anabolic steroids as just a way of, although it's you have to think about the anabolic part is people building muscle tissue. And then there's catalytic. And the function of an enzyme as a catalyst is just what we've already said. It's going to cause a chemical reaction. It's going to affect the rate of the reaction, usually speeding it up or somehow modulating it. So here we see something that is maltose. And this is actually this squiggly line that looks like it's just a bunch of ribbons. This is really more like what a protein would look like and we'll look at this later on. So, but this is showing that up here you can see maltose, a disaccharide, two sugars that are combined by this glycosidic bond, are going to be broken down by an enzyme called maltase into its components, which are these two monomers or monosaccharides of glucose. Proteins are made of monomers. Speaking of monomers, and the monomers of proteins are these right here called amino acids. Again, now we're getting another view of the importance of these different functional groups. So this amino acid always is going to have some basic structure that's exactly the same in all amino acids. And you can see it here. There's first a central carbon, and then attached to that central carbon is usually a hydrogen here, and then some side chain. Now this side chain, we'll look at some of them in the next slide, but this tells us which amino acid it is. But all amino acids have this, and then on one side they have this carboxyl group. On the other side they have this amino group, and that is an amino acid. Now, amino acids, there's 20 of them that are found in proteins. Some of them are, and I see if I can make this a little bit bigger, I'll try. Some of these amino acids are ones that our bodies can make themselves. Some of them are ones that we absolutely need to get in our diets from eating high protein foods. Some of them, the blue boxes around these are showing what those R groups would be. So that's what really determines which one of the 20 amino acids we're looking at in that larger protein. So you can see that some of them, depending on what they have here in these boxes, these R groups, some of them are non-polar aliphatic R groups. So see how much you know now and how much you learned already. So you know that it's a chain, right, aliphatic. It's nonpolar, so most of what you're going to see in here is going to be carbon and hydrogen, like hydrocarbons. And then let's find any other, that's kind of, oh yeah, then there's another nonpolar group, so I'm going to go to the opposite side here. These also are side chains, but notice that these are aromatic, because they have hydrocarbon rings. So these are cyclic structures, and all of them have that. So these three, which are actually pretty common ones, we'll hear in a lot of places, but this is phenylalanine and tyrosine and tryptophan, which some of you may recognize as being one that's associated with a feeling of sleepiness. So these are all nonpolar, these are aromatic R groups, but every single one of these on both sides of this chart have that same basic component of a carbon in the center with a carboxyl group and an amino group on one part of it, and then usually a hydrogen. Well, almost always hydrogen. Maybe I'll even go one more step, always a hydrogen. Okay, so there we go. Let's keep going. Oh, sorry, so those are the nonpolar ones. Notice that there are other types that are polar. Some are polar, but they don't have a charge on them. So notice that in this group we see things like the OH, the hydroxyl functional group, which you know likes to interact with water. So these are the more uncharged but still polar. They'll still interact with water. And then there are some which do have a charge on them somewhere. So these three here have positive charges. These two here have negative charges. You can see that right there. And that's it. So these are all the 20 different types of amino acids. Amino acids, just like, this is the challenge here in looking at biology and looking at the biochemistry, is that there's different ways to talk about all of these things. You can get there's symbols and names and all of that. So when we're looking at individual, if we were going to draw all of the amino acids in a protein and just put them, kind of line them up the way they'd normally be, we could write out each of their names, like valine. But there's also abbreviations for them. And so the V would be a shortcut for writing valine, or sometimes VAL. So it just shows you that this one's a little more cryptic, though, because aspartic acid, which is an amino acid, you can either put this, ASP, which sounds more similar, or D, the letter D stands for that too. As far as essential amino acids, we have to get these in our diet. So these three, in particular, we've got to get into our food supply somehow, which is, these are the ones that somebody who is a vegetarian or vegan has to work hard to be sure they're getting foods, plant-based foods that contain these amino acids. I got a little too big on this one, so now I'm going to reduce the size just a little bit. There we go. So guess what? These units of the amino acids are going to join together and form these covalent bonds through this process of dehydration synthesis. And when they form this bond, so this water molecule is going to get kicked out, you're going to see this is the result, where on one of the amino acids that you'll see that carboxyl group, will just see the carbon with the double bonded oxygen attached directly to the nitrogen or the amino group on the one next door. This is a peptide bond. A polypeptide is another name for a whole bunch of amino acids in a chain, all linked together by these peptide bonds or peptide linkages. And most proteins are polypeptides. You'll see many of them put together. They can sometimes will have something called a prosthetic group. So if you think of a prosthesis in a lot of medical terminology is looking for could be something someone has to, if they lost a leg, you could call that a prosthetic limb. In this case, though, what we're looking at is some addition to the protein that, in addition to what you'd normally see as a prosthetic group, and we're going to see some of them. They talk about how proteins get modified during translation. Stay tuned on that, because we haven't talked about that yet. There's a little video on protein synthesis. It's good to see. And then we go into the different structures of a protein. So we can base a protein structure on these four criteria. First, there's the primary structure, then there's secondary, then there's tertiary, and then there's quaternary. So primary structure is simply all of the amino acids linked together by peptide bonds. But then it gets interesting, because then the secondary structure is caused by hydrogen bonding between parts of this strand that cause it to spiral or to form pleats. After that has happened, then it can go further and go into a tertiary structure, where you see that there's some of those R groups, those side chains, are going to interact. And then finally, some proteins form a quaternary structure, which you can kind of see this by color, a couple of these tertiary structures bound together. So it's a larger, kind of a globular protein. Here it shows the primary structure. You can take a look, you can pause this, take a close look at it. Primary structure is going to be based on which amino acids are in what order on that chain. And they give you an example of when that goes wrong. So if the wrong amino acid is put into the chain when this protein is made, it can cause conditions like sickle cell. Sickle cell can cause the blood cells to form these kind of sickle shapes, and then they are not as effective at carrying oxygen. And I think I'm going to stop there because I'm going to run out of time. I'll continue on the next set.